Our last speaker, we have Professor Ed Boyden. Ed is an MIT graduate in 1999. Where are you, Ed? Ah, okay. He's an associate professor of MIT with appointments in the MIT Media Lab, the Department of Brain and Cognitive Sciences, and the Department of Biological Engineering. The focus of Ed's research is really the development of new technologies, and he's especially known for his work on a technique called optogenetics, which I'm sure he'll tell you something about. These are techniques to allow us to better listen to and control neural activity. Today, Ed's topic is engineering and reverse engineering the brain, controlling the brain to understand it and fix it. Professor Boyden.
All right. Thank you so much for that introduction. So what you've heard today is sort of a wide variety of perspectives in the brain. We know about the brain because it mediates our actions, decisions, our emotions, our social interactions, but we also have learned a lot about the circuitry of the brain, the fact that it's an interconnected network of billions of cells called neurons. And what neuroscience has taught us, as you've heard, is that the circuitry matters. The wiring of the brain results in the transformations of information from our senses, interacting with our memories, all the way out till finally we manifest that information as behavior. Now, one of the things that we've also learned is that these cells are incredibly complicated. So if you look at the cells of the brain, to be honest, we don't even know how many kinds of cells there are in the brain, but they're incredibly heterogeneous. They have many different shapes, they have many different sizes and molecular compositions, and they change in very different ways in different disease states. So just as an example, there is a population of cells called basket cells, an inhibitory set of cells. And these are amongst the cells that are atrophied in diseases like schizophrenia. These cells are inhibitory. They send inhibitory transmitters that will silence downstream cells. But of course, they're networked so that they can also do very complex things like oscillate. Just as another example, there are long projecting neurons, pyramidal cells, that excite their downstream targets and can even go significant fractions of the way across the brain, centimeters perhaps. So how do we get a handle on these? Well, to make things even worse, these cells compute using very high-speed electrical pulses. So these cells exhibit these millisecond timescale pulses of electrical voltage. And those are the electrical pulses that cause them to decide whether or not to transmit information to the downstream neighbors. So to put it in context, within a cubic millimeter of brain tissue, you're going to have 100,000 cells connected by 900 million connections. And those 100,000 cells are each active at the millisecond timescale. So the information processing capacity of the brain is truly astronomical. So one of the strategies that we can try to figure out how this works is to try to control these brain circuits. Can we enter information into the circuits and play back patterns to figure out how these circuits work? If we could turn on one kind of cell, we can see how they influence their downstream targets. If we could turn those cells off, we can figure out what they're necessary for. By going beyond just watching and allowing us to perturb, to find elements in the nervous system, we can try to figure out how these circuits work and how specific components play their roles in initiating, sustaining, or halting different kinds of brain functions and behaviors. So that's kind of our goal. We want to use these tools that we've developed, and I'll tell you about them in a second, to parse out how these circuits work together. There's another reason, though, why we want to learn how to control the brain. And that's because of the enormous disease burden of brain disorders. If you look at the list here of brain disorders that affect humans, that affect perhaps over 1 billion people around the world, very few of them can be treated or cured. And those that have treatments have lots of side effects. And so this is something that we really need to develop new technologies if we want to control these brain circuits in order to make them work better. So if you look at the list here, these are disorders that not only change our life, but also can change our identity even. And so that's why it goes beyond just enormous cost, over $1 trillion per year that is spent trying to help with these diseases, or that are direct losses associated with these diseases, but also because they really change what it means to be human in some ways. Now, in the 20th century, there's a lot of success with pharmacology, and pharmacology continues to be a very, very successful way of treating brain disorders in the sense that it's not invasive, you can take a pill, but we need to figure out where in the brain, where in the brain, if we control it, we can fix brain disorders if we want to develop better drugs. Imagine a drug that would affect one circuit in the brain and not all the others. A drug that just bathes the brain in a substance might affect circuits that you want to fix, but also circuits in the brain that you would like to leave alone. And so it might be part of why brain drugs are so difficult to make. Currently, this is from an editorial recently, they take about 9 years to develop and get approved. They fail 92% of the time in terms of the clinical approval, and the cost for success is about $850 million. So this is a huge need to find better ways to target drugs in the brain in order to figure out how to treat brain disorders, or just to control the brain better directly through forms of energy that you can deliver invasively or non-invasively into the brain. So one of the ideas that we've been playing with is a technology that's going to be known as optogenetics, where basically we would like to be able to control the electricity of neurons, shown at the bottom here, and if we had a way to basically install solar panels in those neurons that convert light into electricity, then we could actually aim light at a cell and turn it on or off, in the same way that you could use a solar cell to charge up a battery. And that might be much more focal than the ability to just bathe the brain in a substance. The other thing is that the brain doesn't feel pain, so you can put optical fibers, like the kind that build the internet and so on, and you can put them into the brain in order to deliver light. So light to electricity, light into the brain. Now we can control cells and turn them on and off using light. Now, to do this, we have to borrow some clues from the natural world. It turns out that all across the natural world, there's photosynthesis and photosensation. And these are mediated by molecules. So what you see here is a single-cell green algae, and in the back, there's a little eye spot that helps it swim around to navigate towards the surface of a pond in order to photosynthesize optimally. Now, if you zoom into that eye spot, there are proteins there, genetically encoded proteins, that when they're hit by light, respond by opening up a little pore and letting charged particles or ions from one side of the cell to the other. So this is directly analogous to the idea that you could use a solar cell to charge up a battery, except now it's done by a biological machine, and it's being done in a way that could be done in an intact cell. This is a protein, so it's encoded for by a little gene in the genome. If we can take that gene and put it into neurons, then we can actually make those neurons light sensitive, was our hypothesis when we began this project many years ago. As it turns out, you have to be very lucky for this to work. There's no guarantee that a plant would be anything like the human brain. There's no guarantee that this protein would express or be non-toxic in the cell. So one of the major parts of this project was to really try to be lucky. I always tell students that's the most important thing if you're trying to build a new tool, is that it's got to work in the messy, intact brain. Surprisingly, the first time we took this molecule and put it into a neuron, the neuron was able to manufacture these little genetically encoded solar panels and install them all over the cell. And when you do that, you can now actually install these little light-sensitive proteins in exactly the boundary of the cell that's important if you want to convert light into electrical activity. And now if you shine light on that cell, you can turn that cell on with light. And so this is great because light can be aimed. We can see we're activating this one cell, but all the neighboring cells in this animation, obviously, are not being affected. So how do you target certain cell types? There are hundreds and hundreds of kinds of cells in the brain. As I mentioned earlier, we don't know for sure how many kinds there are. So we need to use tricks from the field of gene therapy in order to target some cells and not the others. For example, consider that little basket star-shaped cell I showed you in the beginning, the one that's atrophied in patients with schizophrenia. You can actually find genetic regulatory elements and even viruses that will allow you to help target selectively this cell population, so that now you can make that set of cells light sensitive and not their neighbors. Now, as you might imagine, suppose that you want to study a schizophrenia model in a mouse, or maybe someday even use this as a therapy, although that's far off. We could be able to turn on these cells and not their neighbors in order to enter information directly into them. So I want to show you an example of this used for scientific discovery and then an idea as a prototype therapy. And the example I'll show you for scientific discovery uses the system that you heard about earlier, the dopamine system. And one of the questions is, can we figure out exactly which pattern of activity of these cells might be involved with generating feelings like pleasure, or that might drive processes like addiction or learning? So we collaborated with Chris Fiorilla's group in Korea to do this, and they found a genetically targeted way that allows you to make just these cells, the dopamine-producing cells, and not their neighbors, sensitive to light. And that's explained by this acronym up here. We can take questions after the talk for people who want to know what all that means. But the important thing is that he devised a little behavior strategy where there's a little box. Every time the mouse goes to one point in the box, it gets a pulse of light directly into its brain. And every time the mouse goes to this other area, nothing happens. So if indeed a brief activation of this set of cells is enough to make the brain do more of whatever it was just doing, the animal should go over here, essentially working for light. And so that's what we found. The mouse goes here, pokes its nose into a little sensor. You can see the optical fiber implanted in its brain delivering the pulse of light. It's a 1/5 of a second pulse of light. And so the mouse will go to this point, the right-hand one, over and over again, working for light, as it were, and not go to the other one. And so it allows you to prove that brief activation of this set of cells is enough to reinforce whatever the brain was just doing. And now we can imagine all sorts of other things. Can we activate this set of cells and figure out what is happening downstream? But it allows us to do causal perturbations. We can do more than just watch. We can also start to control and figure out whether this set of cells is sufficient to drive a particular pattern of behavior. Now, these molecules come from species all over the tree of life. And so one of the things that has been really powerful is the ability to mine genomes of plants and bacteria and even funguses and critters all over the world to look for molecules that can sense light or that can photosynthesize in response to light. So for example, several years ago, we published a study showing that we could actually find molecules that have the opposite effect. They can turn neurons off. They pump charge the opposite way of the molecules that I just showed you. And so these molecules are quite interesting, and many groups are using them to turn off parts of the brain to figure out what those parts of the brain are necessary for. But you can also imagine someday perhaps prosthetics that would allow you to turn off parts of the brain, for example, right when a seizure is happening, and that might be very useful for treating brain disorders. Now, how realistic is that? Well, 250,000 people already have some kind of neural implant stimulating their brain, mostly cochlear implants, but also deep brain stimulators for Parkinson's disease. So that's a large number of people. Of course, we need to get these molecules into the cells. And so that, in its most simple form, requires a gene therapy, which is not simple. So one of the things that's been happening in recent years is that sort of third and fourth generation viruses have been developed that allow you to deliver genes into cells in safe and effective ways. So for example, just a few months ago in Europe, the first gene therapy in a Western nation was approved by their regulatory body. And even in the United States, dozens of clinical trials are ongoing in order to try and see whether different kinds of diseases can be treated using gene delivery to the human body. Finally, we have to know whether these molecules, which come from bacteria and plants and so on, are safe. And that's something that here at MIT, in collaboration with many groups, including Bob Desimone and Anne Graybiel and others, we've been studying how these molecules work in the non-human primate in order to figure out, in a brain more similar to the human brain, how these are tolerated. And so so far it's looked promising, but of course much more science is required in order to understand how these molecules are treated. Let's talk about a prototype therapy that we've been exploring in collaboration with many groups, and even have started to explore in the context of a startup company that might be able to develop this for human use. And that's the idea that you might be able to help people with blindness. So many people have forms of blindness where the photoreceptive cells, that's these cells in the back of the eye, might be ablated. Now you still have the rest of your eye, but the cells that capture light and transform those light signals into signals the brain can understand are gone. Let's zoom in so we can see this a little bit more carefully. Here at the top are the photoreceptive cells. They capture light and then transform that information through chemical signals through a wide variety of other cells until you get to this layer of cells here, and this layer of cells goes to the brain. So it's almost like a stack of circuitry that's transforming the visual world into signals that the brain can deal with. And in these patients, they've lost the photoreceptive cells that do that first light capture step. Now, light can still get into the eye. So one idea that we've been exploring is whether we could simply take our light-sensitive molecules and install them on a layer of the spared cells, essentially converting them into a camera. So in this collaboration we've been exploring with Alan Horsager at USC and also a startup company, EOS Neuroscience, we've been trying this out in blind mice. Turns out that a lot of the mice you find at the pet stores, you know, the albino ones, for example, are blind just from inbreeding. And so here's a mouse trying to solve a maze, and it's a blind mouse. So the maze has 6 arms. And the goal is to swim through the arms and to get to a little platform right here. Now, this mouse is blind, so it can solve it, but only by doing a brute force search. It has to go down every arm. It's not using vision to solve the maze. We use water because mice like to swim, but they like to sit on platforms more. And so this mouse is really trying to find this little platform so that it can hang out. So these mice are smart. They eventually solve it, but they're not using vision to do it. This mouse, several weeks beforehand, was blind, but got 1 dose of a gene therapy vector that encodes for this molecule, and now it can navigate directly toward the platform. In fact, it can do it in certain conditions as fast as a mouse that's seen its whole life. And so that's really useful and valuable. It doesn't prove that these mice have conscious vision the way that a human might, but it does tell us that the mice can make cognitive use of the visual information, and that's a step forward. Now, for the central nervous system, as I mentioned, many people have implanted brain stimulators. About 80,000 people have brain stimulators implanted to treat Parkinson's disease, for example. And over the last several decades, people have been exploring this. In collaboration with Cliff Fonstad's group here in electrical engineering, we've been trying to figure out if we can do this with light. And one strategy has been to make microfabricated devices that guide light down very thin waveguides, like optical fibers, but much, much smaller, and then can deliver light to various targets deep in the brain. Essentially, light goes down these little waveguides, and then a mirror kicks the light out at 90 degrees once you get to your target. And then we figured out a way to microfabricate these in 3 dimensions. So these are microfabricated devices, but they're macro scale, even as though they're micron precise. So now we can play back activity through a projector essentially coupled to these fiber arrays or waveguide arrays, and you can play back an arbitrary pattern. For example, could you dial information in to the auditory system or the motor system or the sensory systems of the brain in order to replace lost functions that require detailed computational input to the brain? Or here, for example, we're playing back the pattern MIT, which it's not quite clear what the brain makes of that, but I figured this audience would be receptive of us having tried at least. We've also been trying to figure out how to equip these three-dimensional arrays with lots of brain recording sites. So what you see here are electrode arrays that have hundreds and hundreds and hundreds of little tiny metal pads that can pick up electrical signals from the brain. And we can also manufacture these in three-dimensional patterns. So a cubic millimeter brain tissue, as we started off the talk with, has about 100,000 cells. We're not quite at 100,000 electrodes per cubic millimeter yet, but we have been able to build three-dimensional scalable microfabricated systems that might be the basis for future generations of brain prosthetics. In fact, one thing we've been thinking about is this concept of the brain coprocessor. Imagine a machine, and I've showed you many of the prototypes of the building blocks, which can record information throughout neural circuits. It can then compute the information the brain needs, for example, to shut down a seizure. But you could also imagine, for patients with Alzheimer's disease or complex disorders, the ability to play back activity that the brain might need, even memory or cognitive kinds of information, and then use those three-dimensional devices that I showed you to beam light into 3D patterns in order to enter information in the brain to replace those lost functions. And in between can be a computer, and this might be a way that we could study the brain in animal models, but also potentially to pursue these in humans as ways to restore brain functions. Now, what about building brains? This is something we've started to explore, and this is a collaboration with Utkan Demirci's group here at MIT. And the basic idea is, if we really want to understand the brain, we should be able to synthesize brains from scratch. Richard Feynman once said, "What I cannot build, I do not understand." But there's also just the question of whether we could ever build full replacement parts for the brain. Now, tissue engineering has been around for a while, and neuroscience is also, as you've heard throughout the day, an incredibly fascinating and dynamic field. And so our two groups decided to team up and see if we could build three-dimensional brain structures using stem cells and neural precursors and assemble them from scratch. So how do we do this? Well, just like the microfabricated 3D structures for brain interfacing that we talked about earlier, we decided to borrow a cue from the fields of computer science, electrical engineering, and material science, and that's photolithography. How do you shine light on a solution and turn it into a solid? So what we do here is we start with a little piece of glass and flow in a liquid, and the liquid contains stem cells or neural precursors. Then we can shine light onto that liquid and cause it to polymerize into a solid, trapping whatever stem cells or neural precursors that were there into that point in space. We can pattern the light by having a mask or a projector or any one of a number of other ways, essentially making a display that allows us to aim light at certain areas of this growing brain tissue and not others. And then we can repeat this. So over time, we can build up a three-dimensional brain structure, building it bit by bit, or even changing the cells that are deposited at each time, right? Because there's many kinds of cell in the brain, and ideally you could you could specify different cells at different points in three-dimensional space in order to build the complexity of brain circuits. So this is early days, but we have shown some things that can be done. One thing we've shown is that we can actually lock cells into little cubes of this polymer, and then we can have little highways so that the cells can extend processes and link up. We've also shown that these technologies can preserve cell types in realistic ratios as you would find in a normal brain. The excitatory to inhibitory ratio, the neuron to glia ratio, and so on, at least a first order, are similar to what you might expect in an intact brain. Now we're engaged with many downstream processes. Can we do this with human-derived stem cells, for example? You might have heard last year about induced pluripotency stem cells, where you can take a cell from a human, convert it back into a stem cell, then you can convert it into neurons of different kinds. Can we use these technologies to build up vertically these kinds of brain tissues with different cells at different points? In the short term, I think one of the payoffs will be the ability to study the brain. How does it wire up? But also, you can imagine taking a cell from a patient, making a little mini brain that has the same genome as that patient, although, of course, the cells will not be exact replicas. You might imagine this being useful for doing personalized medicine. Could we build little mini brains and then try out different drugs on this brain in a dish before we try them out on a human patient? Maybe that could accelerate the ability to diagnose or personalize medications for people. In the long term, of course, we're very interested in understanding the principles of brain building. Could these eventually be replacement parts for the brain? And what are the principles of doing these kinds of circuit buildings from scratch? We try to give away tools to nonprofits and research institutions for free whenever we can, but also we've been working at an academic level with the Sloan School here to try to teach courses that help people get their ideas out of the lab and into the world. So this fall, for example, we'll teach for the 7th time a course, Neurotechnology Ventures, that I teach with Jos Sponsen, which is aimed at trying to help people get their ideas out of the lab and into the world. And many of the students have actually started companies based upon these projects that they do in the class. So we're very excited about this whole idea of translating neurotechnology out into the world, both to accelerate researchers. Over 1,000 groups around the world use technologies that I've showed you today that we initiated, but also to get new ideas that help build economic growth and that accelerate the ability to get brain technology out into the world. So one of my hopes is that a neurotech valley will start to emerge, although there's not really valleys around here, we can still call it that, I guess, where we can help people get these ideas and build a community that allows people to innovate and start companies and get ideas out of there. If you remember, one of the premises here was that there's a lot of need in brain technology to treat people. 9 years, $900 million, and a 90% failure rate for current brain drugs. If we can help people innovate and get the new models for innovation out into the world, we can help make this world a better place. All of this is a vast collaborative effort with a huge number of people at MIT and around the world. There's not time in the remaining 9 seconds to acknowledge all of them, so maybe we'll close on this slide, and I thank you for your time.